Hello students, welcome to another lecture of pharmacology, and in today's lecture we will be studying about the receptor theories and the classification of receptors. So let's start. So talking about the receptor theory, so after studying quantitative aspects of drug action, Clark in 1937 he formulated a theory of drug action which was based on the occupation of receptors by specific drugs, and that the frequency or the pace of a cellular function can be easily altered by the interaction between these receptors and drugs. He basically perceived that the interaction between the two species, one being your drug and the other your receptor, it can be governed by the law of mass action, and the effect is mainly the direct function of the drug receptor complex which is formed. And subsequently it has been realized that occupation of receptor is essential but not itself sufficient to elicit a response. The agonist must also be able to activate the receptor. Now the ability to bind with the receptor is termed as affinity, and the capacity to induce a functional change in the receptor is termed as efficacy. Now receptors are basically regulatory macromolecules which are mainly protein in nature, though nucleic acid may also serve as a point of receptor. Right? Hundreds of receptor proteins, they have been isolated, purified, cloned, and the primary amino acid sequence has been worked out. Receptors come in many types, and in details we will be studying about the types of receptor in our coming lectures, but they can be broadly classified into two types, one being your intracellular receptor and the second being your cell surface receptor. Intracellular receptor, as the name suggests, is found inside the cell, and the cell surface receptor is found in the plasma membrane. Now talking about intracellular receptors, they are mainly proteins which are found inside the cell, mainly in the cytoplasm or the nucleus. And cell surface receptor, they are membrane bound proteins that bind to ligand on the outside surface of the cell. And in this type of signaling, the ligand does not need to cross the plasma membrane to show its effect. Then comes your action effect sequence. Now drug action and drug effect are mainly used for the same phenomena mainly, but they are not synonyms, right? So talking about drug action, it is the initial combination of the drug with its receptor resulting in a conformational change in the receptor, or the prevention of conformational change through exclusion of the agonist. Meaning that in case of agonist, conformational change will occur in the receptor and there will be response, and in case of antagonist, no conformational change will occur. Now talking about drug effect, it is the ultimate change in the biological function brought about as a consequence of drug action through a series of intermediate steps. So there are multiple steps, and through those steps action occurs. Receptor subserve two essential functions, one being the recognition of the specific ligand molecule and the second being transduction of the signal into a response, right? And accordingly the receptor molecule has a ligand binding domain and an effector domain which undergoes a functional conformation change and shows its effect. These domains, they have now actually been identified in some receptors, and in some they are still unknown. So thank you so much dear students. This was all in today's lecture. Today we studied about the receptor theory and the different types of receptors through which the action of drug occurs. If you have any queries, any doubt in today's lecture, please feel free to reach out to me. Thank you.